can get inflammation, inflamed. Okay? And that is tonsillitis. Okay, right. So inflammation. And sometimes if that happens too much, then you remove the tonsil. The reason is, although tonsils are very important, but you have many other organs, right? Lymph organs. Like you have so many lymph nodes in your body. You have other organs where you have plenty of follicles. Okay? So not necessarily you have to keep it. So its removal will not, do any harm. This is the real picture of the cortex part, this part. So you see the follicles, the darker round structures. So those are the follicles. Now we will talk about the lymph organs. Organs are larger than the lymph nodes, and they are responsible for immunity, but they have other functions too. That's what I mentioned. Now, spleen. The function of the spleen is, it is a site of lymphocyte proliferation and surveillance and response. Immune surveillance and immune response. Now, proliferation means increasing number. So, lymphocytes multiply inside the spleen. So, the number of lymphocytes increases. Now, how many new lymphocytes should be produced? That is determined by the need. If your spleen thinks that need to produce more, it will produce more. If no, it will not. So that's the surveillance. It will monitor the antigens. And depending on that, it will respond. Now, another function of the spleen is destroying the old blood cells. So this is not immune function, destroying the old blood cells. First one is immune function, proliferation of lymphocytes, right? Surveillance and response. Second one is removing the old blood cells, and that is the cleanses of the blood. You already know that spleen and liver, these two organs are called what? Yeah, graveyards. Remember that? Graveyards. So we already know that inside the spleen, the blood cells are destroyed. Inside the spleen, you have lymphocytes, as well as you have macrophages, huge number of erythrocytes, of course, because red blood cells are destroyed there. In the spleen, you have two distinct areas. If you cut the spleen and see inside, you can, without microscope, if you cut the spleen and see inside, you can see the white and red areas. Okay? So white areas are called white pulps, and red areas are red pulps. So you have white and red pulps. Now, why one area is white, another area is red? Inside the spleen, if you see, there are arteries like this. Okay? Those are called central arteries. Central arteries. Okay? Around the central arteries, the area is white. So around the central artery, the area is what? White. So that is white pulp. Now, in some areas inside the spleen, you have sinuses. Sinuses are the cavities. Okay? So these are the sinuses. And these sinuses get filled with blood. So these are venous sinuses filled with blood. So when you see this area, it looks red because the blood accumulates inside the sinuses. So this is the red pulp. Okay. So around the central artery, you have white pulps. Around the in the area where you have the sinuses, those are filled with blood. You have red pulps. Look red. Okay. In this picture, you see the areas shown by blue color. That means venous blood. Okay. Blue color means what? Arterial or venous? Venous. In the textbook, remember, I told you before. In the textbook, it is easy, right? You see in the model, red blood vessels are what? Arteries. And blue blood vessels are what? Veins. But in real, body, blood is always red. Blood is always what? Red. So that is just for you to know in the picture, just for you to remember. This is artery, this is vein. But that means this blue doesn't really look blue. It is red, right? Because blood is there, accumulates there. So that area looks red. That's why red pulp. Is that clear? So I don't like those pictures. They are far from real things. Okay, so that is the spleen. Now, thymus, another lymph organ. Thymus is interesting. In early stage of life, if you see the thymus of an infant, it occupies almost whole upper part of the chest, whole upper part of the thorax. So it is very big, large. Okay, and gradually it gets smaller. Smaller, and in adult it becomes like this. So why is that? Thymus is very big in early stage of life because it takes years to develop our immunity in the body, right? Immunity develops what? Slowly, gradually, right? When you get exposure to any antigen, your body creates one type of immunity, right? Then another type of antigen, your body produces another type of immunity, right, in your body. So, immune system gets matured or develops slowly. But in early stage, immune system is very weak, right? So the thymus has to work much more. Make sense? So that's why it is big. Then when the use becomes less and less, it shrinks. Okay, gets smaller. Okay, now thymus has lobules, lobes, and those are called thymic lobules, contain cortex and medulla. So in the thymus you have lobules or lobes, and inside the lobe you have cortex and medulla. And in the cortex you have densely packed lymphocytes and scattered macrophages. Densely packed lymphocytes are actually like this, the follicles. I mentioned that, packed lymphocytes. So in the cortex, you have follicles and also macrophages scattered. And in the cortex, you have, in the medulla, you have a structure that is called Hassel's corpuscles. Thymic corpuscles. Hassel's corpuscles, the function is not yet clearly understood, but we see in the medulla Hassel's corpuscles or thymic corpuscles. Some people reported, or some research reported, that thymic corpuscles are the areas where the lymphocytes get matured inside the thymus, get training. You remember some lymphocytes go to the thymus, right, and get out, become as T lymphocyte. So some scientists, they believe that Hassel's corpuscles are the structures where these structures are needed for maturation of T lymphocytes. Here you see the cortex part you see is dark and medulla is less dark. Why? Because in cortex you have packed lymphocytes. That's why the area looks dark under microscope. Many cells, lymphocytes. In the medulla, you see that round structure that is a Hassel's corpuscle. Important for the maturation of T cells. Tonsils. Tonsils are palatine tonsils, lingual tonsils, and pharyngeal tonsil. Based on the location, they have been named on their locations. Palatine tonsils are located behind the soft palate. So near the palate, that's why called palatine. Lingual tonsils are grouped at the base of the tongue. The tongue means lingual, you know that, right? Lingual means tongue. So lingual tonsils at the base of the tongue. And pharyngeal tonsil is located or attached to the nasopharynx. Nasopharynx. That's why called pharyngeal. Okay. So here you see the locations and histology of a tonsil. In tonsil, you have crypts, folds called tonsillar crypts. And why those crypts are present in tonsil? It is said that the fluid containing lot of antigens can be trapped inside those crypts, can accumulate inside those crypts, and then they will be taken into the thymus and tonsils and will be destroyed. Okay? So in these folds, plenty of antigens get stuck, like, you know. These are called crypts. Okay. So the fluid with lot of antigens accumulate there. We know that lymphocytes are present in huge amount in the tonsils, lymph nodes, thymus, spleen, but also in few other structures of your body, the lymphocytes are present in huge number. One is in the late part of your small intestine, distal part or late part of the small intestine. In the wall of the small intestine, you have aggregated lymphoid follicles. These structures, many aggregated in the wall of the intestine, and those aggregated lymphoid follicles are called Peyer's patches. Peyer's patches. Inside the appendix, you also have the aggregated lymphoid follicles. Those follicles are heavily filled with lymphocytes. You know that. Now, just think that why you need those aggregated lymphoid follicles in the wall of late part of small intestine. Because if you just think that which way the antigens can easily enter into your body, through your respiratory, right? Because you are taking the air 24 hours from outside, right? Another is what? Food. You are taking food, right? So the food goes to the intestine, right? So that is another way the antigens can enter into your body from the food, right? So that's why in the wall of the small intestine you have what? Those follicles. Makes sense? Filled with lymphocytes. So if any antigen, from the food stuff tries to enter into your body, they will destroy them, right? So those areas you need more guards. I mentioned that, right? Where in the border you need the security post, check post. So like that. Lacteals. Lacteals are the lymph capillaries. Where? In the small intestine. So if you see the wall of the small intestine, you have villi. Villi are finger-like structures in the small intestine. So these are, some people say villi. Those are finger-like structures in the wall of small intestine. And your food stuffs are here. Proteins, carbohydrates, fats. Food. And this is the wall, okay, of the intestine. So from the wall, you have finger-like structures called villi, and you have microvilli here too. Anyway, so the blood capillaries enter into the villi. So these are blood capillaries as well as lymph capillaries. So this is the lymph capillary, the green one, okay? So both blood capillaries and lymph capillaries are there. Now, the blood capillaries can absorb the proteins and carbohydrates easily, because proteins and carbohydrates are small molecules. Proteins and carbohydrates can easily enter into the blood through the blood capillary wall. Proteins and carbohydrates. Okay? Those nutrients can easily enter into the blood capillaries. But fat molecules are larger. Fat molecules are what? Bigger. These are the fat molecules. Okay? Fat. They cannot easily enter into the blood capillary because in the wall of blood capillary you have very small holes. Fat molecules are larger than that. So they cannot easily get into the blood capillaries. But in the wall of lymph capillaries you have larger holes. So what happens is, see, the fat molecules enter into the lymph capillaries instead of blood capillaries. Is it clear? So fat molecules enter into the lymphatic system, lymph capillaries. Okay, so that's how the nutrients are absorbed, and these lymph capillaries in the villi are called lacteals. Lacteals. Okay. Lymph capillaries in the intestinal villi or mucosa. Okay. Now you may think that nutrients should enter into the blood, not into the lymph, right? Proteins, carbohydrates, fats should enter into the blood. But it is not a problem because we know that finally lymph is given to subclavian vein, right? That means what? Into the blood. Subclavian vein means blood, right? So the lymph is going to the blood. That means the fat molecules, those are entering here into the lymphatic capillaries, will eventually enter into the blood. Makes sense? Will get mixed with the blood. So it is not a problem. Okay? So fat absorption is the function of the lacteals. Okay. You see here, this is the wall of late part of the small intestine. You see many follicles. These are filled with lymphocytes. So those are called Peyer's patches. Okay. So basically, what you have learned today, first is you have learned the parts, right, of the lymphatic system. You know the lymphatic vessels include the capillaries, collecting vessels, right, trunks and ducts. Ducts are the largest, right? How many ducts you have? 2. The larger one is what? Thoracic duct. And that receives lymph from which part of the body? 3/4. Very good. And right lymphatic duct receives from 1/4. Which 1/4? Upper right quadrant. Make sense? Okay. And yeah. Then lymphocytes are the main what? Warriors, right? 2 types of lymphocytes. What are those? T. Why one is called T, another B? Because they are trained differently, right? They get their training in different locations and they are trained differently, right? One learn to do what? Secrete antibodies, right? Another to engulf. Okay? Phagocytosis. So that's how they are different. Then you have learned about lymph nodes, right? Structure of lymph node, function of lymph node, tonsils, thymus, spleen. Briefly, we went over those, right? If I ask you, red pulps, white pulps are present in what? In the spleen. If I ask you, okay, in which structure you have sinuses, red pulp or white pulp? Red pulp, right? Because those sinuses are filled with blood, right? That's why that area looks red. Around central arteries, you have white pulp, right? Hassel's corpuscles are present in which organ? Thymus. Right? T cells are, we think that T cells are trained by the Hassel's corpuscle, right? So must be in thymus. Tonsils, 3 types of tonsils.